Hey there, science fans. Today we're diving into the fascinating world of bacteria. Those tiny organisms that are everywhere. But how do we tell these microscopic critters apart? That's where gram staining comes in. It's like magic, but with science. Gram staining is a crucial technique in microbiology, helping us classify bacteria into two major groups, gram-positive and gram-negative. Think of it as dividing our bacterial buddies into two teams, wearing different colored uniforms. This distinction is not just about color, it reveals fundamental differences in their cell walls, which dictates how they react to different antibiotics. Before we start, a little history. This ingenious staining method was developed by Danish scientist Hans Christian Gram way back in 1884. He was examining lung tissue from patients who had died of pneumonia. He noticed that some bacteria retained the crystal violet stain after treatment with alcohol, while others did not. This observation led to the development of the Gram stain, a technique still widely used in microbiology labs today. Pretty neat, huh? Now, you might be wondering, why is it so important to differentiate between gram-positive and gram-negative bacteria? Well, my friends, this simple stain provides crucial information that helps doctors choose the right treatment for bacterial infections. Knowing the gram stain characteristic of a bacterium can help guide antibiotic therapy, as different antibiotics are effective against gram-positive and gram-negative bacteria. So, let's put on our lab coats and delve into the colorful world of gram staining. Every good magic trick needs its secret ingredients, and gram staining is no different. We have 4 key reagents, each with a specific role to play in this microscopic color show. First up, we have crystal violet, our primary stain. This purple dye is like the initial flood of color, staining all bacteria with a vibrant violet hue. Think of it as the base coat of paint. Next in line is iodine, our mordant. Now, mordant might sound like something out of a Harry Potter book, but it's simply a substance that helps fix the dye onto the bacteria. It forms a complex with crystal violet, making it less soluble and more likely to stick around. Think of it as the glue that makes the color stick. Now for a little drama, we introduce ethanol or acetone, our decolorizing agent. This is where the real action happens. It's like a magic eraser that selectively removes the color from gram-negative bacteria, while gram-positive bacteria hold on to their purple coats. The suspense is killing me. Finally, we have saffron in our counterstain. This pink dye steps in to stain the decolorized gram-negative bacteria, giving them a lovely pink or red outfit. It's like giving the other team their jerseys so we can tell them apart. Now that we've met our star players, let's get down to business. The first step in gram staining is preparing a bacterial smear. Imagine spreading a thin layer of peanut butter on a slice of bread. That's kind of what we're doing here, but with bacteria instead of peanut butter. We take a tiny drop of our bacterial culture and spread it thinly on a clean glass slide. This ensures that we have a single layer of bacteria to observe under the microscope. It wouldn't be much fun if they were all piled on top of each other. Once our smear is ready, we need to fix it. This is a fancy way of saying, let's make sure the bacteria stay put. We do this by gently heating the slide, which kills the bacteria and makes them stick to the glass. Think of it as lightly toasting our bread to keep everything in place. Now our bacteria are ready for their colorful makeover. With our bacterial smear prepped and ready, it's time for the main event, the staining process. Now this is where things get really exciting. First, we flood our slide with crystal violet, our purple dye. We let it sit for a bit, giving the dye plenty of time to soak into all the bacteria. It's like giving our bacteria a nice, relaxing bath in purple dye. Next up, we rinse off the excess crystal violet and add iodine, our mordant. Remember, this helps fix the dye onto the bacteria. We give it a few moments to work its magic, then rinse again. Now the real drama begins. We carefully apply ethanol or acetone, our decolorizing agent, watching closely as the magic unfolds. This is like a magic trick. The gram-negative bacteria will lose their purple color, while the gram-positive bacteria hold on tight. After a quick rinse to stop the decolorization process, we're ready for our final step, counter-staining with safranine. This pink dye will stain the decolorized gram-negative bacteria, giving them a contrasting color to the purple gram-positive bacteria. It's like adding sprinkles to our microscopic masterpiece. We rinse one last time and allow our slide to air dry. Now for the moment of truth. Are you ready to see what we've got?